Andrew Bartko from UCSD Center for Microbiome Innovation is going to do the first presentation, and then we'll have a roundtable discussion afterwards.
Great. Well, thank you, Adam, for the introduction. Can you see my screen?
Yep.
Great. Well, thank you. Really excited to have the opportunity to highlight some of the work that we're doing at UCSD to combat the pandemic and reducing the SARS-CoV-2 transmission on campus. So we have a multi-pronged effort on campus that's entitled Return to Learn. I'll just be covering the part of our efforts to combat SARS-CoV-2 from the wastewater perspective, but we do also have contact tracing activities underway, as well as rapid diagnostic tests for people that are symptomatic and asymptomatic as well. So a little bit more about me. My name is Andrew Bartko. I go by Andy, Executive Director of the Center for Microbiome Innovation, and I'm a professor of practice in the bioengineering department at UCSD. So my contribution to the wastewater monitoring activities is really an opportunity to continue. A great deal of my career has been in environmental monitoring for pathogens and hazards. And when I was onboarded to UCSD, it was a welcome introduction to continuing that work throughout my career. With SARS and transmission, there's an opportunity that's really a time-based opportunity to intercept people that may be asymptomatic but infected, and to move them to an isolation or quarantine area prior to them, transmitting the infection to other people that they might be in contact with. So if an individual is simply seeking a test because of some sort of symptomatic response to the virus, there's a significant delay, and there's a lot of people that person would be in contact with prior to them, being diagnosed as a positive for the infection. Although we don't know the timeline or when people begin to shed the virus through their metabolism, we know that it is prior to them developing symptoms. So the opportunity the opportunity that we saw to intercept that timeline was really in monitoring the wastewater from dormitories and other buildings on campus to identify the viral component from that waste stream, and then using that knowledge to advance tests, diagnostic tests to the individuals that occupied that space, prior to them, prior to the infection spreading. So the theoretical timeline is that we can, save days and countless numbers of people that might be infected simply by monitoring that wastewater. So in the October timeframe, October, November, I was fortunate enough to volunteer to be part of the surveillance. And what the surveillance really is sampling wastewater from the manhole covers that encompass the university's buildings. So here's a picture of one of the manholes that we thread a tube down to intercept some of the wastewater that's in that waste stream, collect it for about 24 hours, slowly accumulate a sample using this apparatus. We have currently 100 different sampling sites throughout campus that harvest samples that we then return on a daily basis and run a quantitative PCR assay that is looking for the SARS-CoV-2 viral component. The workflow for that sampling really begins by the sample being transported back to the laboratory. So Once the sample is returned to the laboratory, we extract the DNA and RNA from that sample. We enhance the viral component of that sample and then run a qPCR test. There is another opportunity there, though, with that sample, in that it also has a variety of microbial DNA within it. And those microbial DNA come from the environment, but also from the gut microbiome of the donor of that sample. So that is the opportunity to analyze the sample in a greater detail, and to understand not necessarily how it's modulated by the virus, but also opportunities to see if there are other components within the environment that might be anomalous within the sample itself. So we take that sample, we run it and generate sequence data, run a quantitative PCR, the simple kind of standard quantitative PCR assay. We then map those samples to where they occurred or where they were sampled from campus, and we have a geospatial map of what buildings are infected, or what buildings have occupants that are infected and which ones do not. So here is our dashboard that was I pulled from our online dashboard a couple days ago. And what you can see here is in the darker gray are all the actively monitored buildings on campus. We started with dormitories and residence halls to monitor them because they had the greatest population, and also you know, had more capacity than some of the research facilities that we have that are in the lighter gray. All the red highlighted buildings are buildings that had tested positive. The wastewater was tested positive for the SARS-CoV-2 virus. What happens after this is, you know, broadcast to all the building occupants of those facilities. You know, the recommendation is to have a rapid diagnostic test, either a nasopharyngeal test or a saliva test, to determine whether they're symptomatic or whether they're infected or not. They don't necessarily know that they're that they're infected prior to the test. So I saw this in action, and it was really a gratifying event to actually pull a sample from a dormitory wastewater stream. That sample tested positive. Within 24 hours, the occupants of that dormitory were tested. We have identified 2 individuals within that dormitory that were then offered to quarantine in some of the buildings that you see over here on the east side of campus is where we offer that quarantine space for them to recuperate. So that was an end-to-end positive experience for me to see the power of this particular surveillance tool. On average, if you look at the campus, we're about 10 times less infectivity rates are about 10 times less on campus than they are in the surrounding communities where we don't necessarily monitor for wastewater. So that's an amazing capability that we've spun up rapidly to really fight the pandemic on campus. So as I mentioned before, there's an opportunity as well. We don't necessarily have to just rely on quantitative PCR. We've ran several different other studies that looked for correlations between a SARS-CoV positive sample and how that SARS-CoV-2 sample influences the microbiome, or the bacteria that is within that ecosystem. And what we found is that the virus does modulate the composition of the microbial communities within a variety of different sample types: nose, forehead, stool, and in the built environment, the inside floors, where you might have respirable particles shed from people and landing on the floor. So high performance computing really enables this for us, in that we can generate these types of phylogenetic trees and look for correlations between different types of sample types. So this phylogenetic tree that I'm showing here on this plot is really enabled by that ability to plot these sequence data quickly and efficiently. And earlier this or late last year, about 4 or 5 months ago, we were able to actually plot about 1.4 million different unique sequences on one phylogenetic tree. And what that enables us to do is see over the horizon for how trace amounts of virus or trace amounts of perturbation within that ecosystem can be visualized in a plot like this. So, 1.4 million species or organisms within that sample is orders of magnitude more than what is typical for these types of phylogenetic trees. And then finally, how we're going to leverage that additional information that comes from viral-induced perturbations within the microbiome community is really not about the SARS-CoV-2 virus pandemic, but also looking for other types of anomalies within the landscape that we're working within. So one of the big problems that we have as a species really is we have all these built environments, building owners. So the example that I'm showing here is, for instance, if you're the owner of the Empire State Building or a tenant within the Empire State Building, you're probably asking yourself, when can I fully occupy my space again? When can I, be back to a new normal? And although we don't know when that's going to be, we have the tools in place and feasibility shown with what we're doing with wastewater to impact or to provide some informed information that the building owner can use. So the notion here is that we can monitor each floor or each kind of a division within a building, their wastewater streams from their facilities on those floors, capture those samples, sequence them using our standard pipeline to look not necessarily for quantitative PCR, but also for the microbiome sequencing that happens, that coalesces with the waste streams of the people occupying that building. That information is processed with our bioinformatics tools to look at the phylogeny of bacteria within that space. And through looking at a time series of that information, we can see how that phylogeny changes over time through a longitudinal study. If we find that there is anomalies or things that we can't necessarily explain within the changes that we see within the microbiome, then we can alert the building or the floor owner, the tenant within the Empire State Building, to tell them, hey, we have a positive for a different virus or something else. Send your people to get tested that occupy that floor, and then quarantine them from the building for a period of time where they can either become symptomatic and treated, or that symptomatic condition goes away. So that's the notion of where we're going next. And with that, I will turn it over to all the acknowledgements. The Night Lab is key for UCSD's Return to Learn program with wastewater surveillance and all the genomic sequencing that we do. And with that, I'll turn it over. Also, huge thanks to Panasas for our collaboration, where we are now enabled to look at massive amounts of data and in a very timely manner. So with that, I'll turn it over to Adam to follow up with this.
Sorry, before you pass there, I did raise my hand because I've always wanted to raise my hand on a Zoom call, but I've been stuck in this hotel for 3 days in Dallas, and we've kind of become a family. Essentially, everyone's pitching in. I got to help a woman in a wheelchair get to a different room because she was stuck there all day freezing, and People are helping out with the food because there's not enough food. It's become a beautiful thing as far as the human connection, which really isn't COVID, but of course we're all talking about COVID at the same time as no power and no running water. A gentleman last night that I got to know quite well now because I've been playing with his boys. In fact, I told my daughter, I'm in a hotel full of families with dogs and kids, and she said, "Dad, you're in heaven. You're in heaven for you." But he had COVID and recently has no diabetes history in his family, and quickly had problems with his pancreas and is now a type 1 diabetic. So we hear in the news all the time that comorbidities affect people obviously in the way that they get it and how it treats them, but the elements that are coming out are things that I don't think we hear that much about in the reports and the news. So are there other things that are of interest to the, maybe to this group or anyone that might view this in the future?
Yeah, that's a definitely a good question and an important one. Andrew, are you seeing, I mean, microbiome, I think the research has really ramped up recently and there's a lot we still don't know about it. How are you seeing, I guess, other researchers at your organization focus on the microbiome and how they can collaborate with you? And this is everyone on the call. I believe you can unmute yourself and you can ask questions or put them in the chat as well.
So just to touch on that, there's a hearty effort with looking for other viral strains of the COVID-19 disease within the university setting. You're right in that microbiome is an emerging, well, it's not necessarily emerging. It's been an established field of research for probably over a decade at this point. But most recently, how the microbiome modulates human health has really stepped up, and the causality is still a bit removed, but the correlations between different microbes and how those microbes influence different types of disease, such as like IBS and Crohn's disease, is well established, and there are therapeutic interventions that are developed specifically for those. The next types of disease states that microbiome is shown to be responsible for are things like depression, schizophrenia, and some types of gut-brain axis diseases have shown correlations, and then also things like NASH and liver diseases as well. So the field is growing. How the field or how microbiome can be used to leverage SARS-CoV-2 ailments or things that Brian was mentioning, I'm not sure. I think it's still a little too fresh at this point to make those assertions at this point. But I suspect
but clearly you'll need more storage to be able to help do that, then, right?
Absolutely.
All right, thank you very much. Sorry, Adam.
Thanks again. And Andrew, you will have to stop sharing for me to start sharing. So this is the last of the sessions. This is meant to be a roundtable discussion, and it's what does hot and cold data mean to you? We hear those terms all the time. And for those of you who don't know me, my name is Marko. I'm the Director of Life Sciences here at Panasas. And I'm trained as a bioinformaticist, but I also held multiple roles as a sysadmin and worked with lots of storage systems, lots of compute, lots of applications. And I only recently came over to the vendor space where I help our customers and potential customers understand our product and help our engineers understand what our customers want. So for roundtable discussion, I just put some key terms that we hear all the time, and I put them in a circle to make it like a roundtable. And I can go ahead and start, but I would appreciate it if anybody on the call would want to join in. I don't know if I can see the chat window, so if someone can see the chat window and relay messages, if people send them, but you're free to come off mute and talk as well. And we hear more and more about hot data, cold data, sometimes in between, like warm or preheated data. As we grow our data sizes, and if any of you were on heard from Quantum earlier, they said they want to keep things for 100 years. We encounter that a lot, and researchers want to keep their data, but how long do they have to keep it? How much is latency? How much does latency affect things? What happens when you want to use your data for AI and ML training data sets, and you can't necessarily wait to get that data back from tape, for example? I've tried copying large sets of data from tape, and it's not really tractable, much less even finding stuff that's on there. So the idea of tiering, I think, logically makes sense. You've got this really fast NVMe or something layer, and then maybe a medium spinning disk layer, and then you move things to tape or the cloud. But we've taken a different approach to that. I know other organizations may be looking at that, so I'm curious what everyone thinks is a workable solution to managing all of the data that's generated in life sciences, and how you can deal with latency issues and single namespaces, and really maximizing the value of all these petabytes, if not exabytes, of life sciences data that have been generated over the years and will continue to be generated without having to move them back and forth onto a small NVMe layer. I'll wait for a little bit and then talk about how we handle that.
I think they're pondering the fact that the round table is not all that round, since you have quite a few more letters on one side.
It's an oval table. Well, I can talk about how we do tiering in a very loose sense of what tiering is. So you can just focus, won't spend too much time on this, you can just look at the left box there. We use NVMe, we use SATA SSDs, and we use spinning disks, but we don't separate those where you have to access the NVMe for your hot analysis, your SATA SSD for somewhere in between, and then the SATA hard drives for long-term storage. This is all transparent to the user. It's all a single namespace. Your metadata goes to that ultra-low latency, fast NVMe. Small files go to the SATA SSD, and large files go to your spinning disk. And then we dynamically manage the data across those for you so that you get large capacity with large capacity storage, but with the performance that you'd expect from a high performance flash tier. So all this is transparent to you. You don't have to configure anything. You don't have to think, where am I going to put stuff on the NVMe? What am I going to do when the scientists fill it up? Do we have to move things back and forth? That doesn't happen with our system. And I have two real world examples. Here's one of a more traditional tiering method. And this is you could find this online. I won't mention who the vendor is, but Genomics England has a hybrid tiered solution. So they've got NVMe, and then they've got an object store. They get about 135 gigabytes a second from that NVMe layer. So that's all flash. And however, it is only 1.3 petabytes as compared to the total available space, which is 40 petabytes. The remainder, that other 97%, that's on spinning disk on object store, which is going to give you probably an expected, but a much lower level of performance. So if you do need to get back to that data, you have that latency issue that you're going to face. And this is becoming more and more of a problem as we move into the AI and ML space. Now, if you were to develop a similar system or deploy a similar system with Panasas at around 40 petabytes, you actually get a much higher throughput performance. You get about 410 gigabytes a second from that entire capacity. So you wouldn't have to worry about moving things back and forth. You wouldn't have to worry about where it is. You wouldn't have to worry about that latency. So not only would your throughput be improved, but all of that namespace would be in one location, and the researchers could access their data at a single expected high performance latency. And in addition to that, I said I didn't mention the vendor. I forgot to get rid of it. I guess we all know who it is, although this is public. We estimate it probably would be about half the cost of this system to go with Panasas over one of these traditionally tiered NVMe object store hybrid solutions. And here's another use case, just in AI and ML, where we did some internal testing. This comes from another vendor who I may not have on here. This information is available online. That's a white paper from another vendor, and they used TensorFlow with an ImageNet dataset to run some AI and ML benchmarks against NVIDIA V100 GPUs. Now, their test is solid. There's no problems with it, but we wanted to compare what would ours look like as compared to an all-flash solution. So our ActiveStor Ultra, this is for a half rack. If we did the test equivalently to how this vendor published it, we would support 42 NVIDIA V100 GPUs at about 1.5 petabytes of raw capacity. To go with that other vendor all-flash array, yes, they would support slightly more GPUs. It wouldn't be a parallel file system like Panasas, and you have about half the capacity and almost certainly several multiples of the cost, because we provide that capacity and performance, but it's spread across the different storage media as opposed to having you pay for all of that high performance flash. So you get twice the capacity, probably about a fifth the cost, and you still support a similar number of GPUs. And 42 V100s is still a ton of GPUs. I mean, that's in hundreds of thousands of dollars of GPUs that you could support in a half rack. So these are some use cases where we look at how tiering can be handled differently, and it doesn't have to be the hard to set up, hard to manage, and seemingly higher performance multiple tier levels when you could have all this integrated in one system. And that's all I have today. I certainly welcome any questions, and Andrew can answer additional questions as well. But I definitely appreciate everybody's time, especially staying the extra, what do we go, almost 40 minutes over.